Welcome everybody to this meeting of the Chemical Consultants Network. The Chemical Consultants Network is a topical group of the American Chemical Society Philadelphia section. And our purpose, our mission is to help people start and prosper their consulting businesses in the chemical enterprise. So we have people from all different areas of technical areas of chemistry, but we also include executives and salespeople and computational chemists and biochemists and pharmaceutical people as well. We would like to request that anybody who is not speaking, and that's going to be only Roman who'll be speaking shortly, please keep your microphone on mute. And the other thing we ask is that if you have questions, please write them in the chat off to the right, and Roman will take questions at the end. And with that, for the introductions, I'd like to turn it over to Dr. John Newport, who has worked with Roman for many years. He's a close friend and is also former chair of the CCN. John, could you please take it away?
Surely, thank you, Keith. It's my pleasure to introduce Dr. Bielski. As Keith said, a friend and colleague. We've worked together for many years. Roman has a master's in chemical engineering, a PhD in organic chemistry. He postdoced with Sir Derek Barton at Imperial College. Derek Barton got a Nobel Prize. Roman is still waiting for the call from Stockholm, but one of these days we hope it will happen. Roman has written many papers, about 50 publications, authored 3 books, has written about 20 patents, some more in the pipeline. I'll tell you a quick anecdote about him. A few years ago, a student in Poland asked his professor a question, and the professor said, "There are only 2 people who can answer this, God and Dr. Bielski. He'll be here next week, ask him." It turned out that God didn't know the answer to the question either, so it was Roman who held the day. What else can I tell you about him? Currently, Roman partners with me in Chemventive in Biosynthon. He's an associate professor at Wilkes. He is a master of many things, and it's my pleasure to introduce him tonight, and he's going to tell us all about click chemistry. Roman, it's all yours.
Thank you very much. I have some problems to share a screen at the moment, but I'll try to get that. Thank you very much, John and Keith. It was very kind. The words were too kind, but I will accept them. My fellow chemical consultants. The title says that it's a novel tool. It's not that novel, but it's very important, and I'm very happy to be able to present it to you, particularly because I believe some of you don't know the click chemistry, and it's a major development. I'm considering becoming a politician, therefore I'm considering starting to like my voice. Therefore, I'll talk a lot. I'll talk a lot before entering the real click chemistry, but I hope I'll give you an interesting perspective on click chemistry and its place in organic chemistry. So let me start you with the beginnings. It all started. Well, I don't know how to move to the next slide, and it's not. Here, the beginnings. So it started in 1828. Friedrich Wöhler was trying to make ammonium cyanide, that's its structure, and in the process he heated the product, which probably he synthesized, and he ended up with this very compound, urea. It could not have happened. That was clearly impossible. The reason is that there was this concept of vis vitalis, vital force, compounds produced by living organisms, trees, plants, whatever, are produced as a result of the special force. And inorganic compounds cannot be transformed to organic compounds. And it was the end of this wrong theory, but the final end happened some 15 years later when Kolbe synthesized acetic acid from carbon disulfide. Interestingly, Kolbe was the first to introduce the term synthesis. And so the organic synthesis started then, and soon we learned that we can isolate compounds from natural sources, determine the structure, and eventually synthesize them. And that's very important. Some 14 years ago, in 2006, Samuel Daniszewski, a wonderful, great chemist, offered a perspective in Journal of Organic Chemistry where he makes a point that if you take randomly compounds you can think of and synthesize them, let's say 1 in a million will be biologically active, will act as a drug. But if you take natural compounds, then maybe 1 in 10,000. The probability of hitting the jackpot are much, much better. Why? Well, all the compounds, even the terrible toxins, must have gone through the very tedious and long selection, better than the one the FDA offers. Millions of years, these compounds were in presence of other enzymes, and nature selected eventually compounds which do not affect enzymes essential for various organisms. So my point is, natural compounds and the derivatives are essential for successful pharmaceutical industry. The 20th century was the century of natural products and their derivatives. We talk also about the natural product-based medicines or drugs. Let me give an example of insulin, which is the polypeptide consisting of, I think, 2 chains, one's about 30, the other one about 20 amino acids. It was isolated in, I believe, 1921 was the first paper. The Nobel Prize for these 2 Canadians was in 1923, 2 years later. Structure, I think Sanger published his paper on the structure of the insulin in the 1952, maybe 53. Sanger got Nobel Prize in 1958. Total synthesis was performed by these Americans and Chinese, 2 independent groups. The 3-dimensional structure was determined by X-ray crystallography by Dorothy Hodgkin, who got a Nobel Prize in 1963 for developing this modern X-ray crystallography. And just little different chemistry, but still wonderful chemistry. Rosalind Yarrow got a Nobel Prize for radioimmunoassay, competitive radioimmunoassay. And the same way, variety of compounds were. The structure was determined, and then they were synthesized. Carbohydrates, steroids, terpenes, and so on. At the moment, I'm writing a paper on the 50 years of the Akhmatovich rearrangement, which I was involved in. And it's the method to make 6-member ring system with 1 oxygen. Wonderful method. And I wanted to show the most sophisticated, synthesized, almost synthesized. By the way, Nicolau, who is the author of the synthesis, synthesized almost all the components, all the units. But the final synthesis of everything didn't happen because the richest country in the world cannot afford it. There was not enough money. Anyway, look at this toxin, phenomenal compound. 32 rings, 98 stereocenters, 28 tetrahedropyranal rings, 6-member ring with oxygen, 1 geometrical site, this one. And lethal dose, LD50, is 50 nanograms per kilogram. So 1 person will be killed roughly with 4 micrograms. 4 milligrams is enough to kill 1,000 people or so. Fantastic compound, but this very compound was practically synthesized, the crown prince of synthesis. And let me make a point. Synthesis will continue. There are new fantastic developments in the synthesis and in synthesizing chiral compounds having chiral centers. But certain events allow to look at organic chemistry as producing new, very, very important avenues, new avenues. First one is bioorthogonal chemistry. Carlo Bertozzi in California, first at Berkeley, now at Stanford, came up with this phenomenal concept, which is exceptionally fruitful. Any chemical reaction that can occur inside of living organ systems, without interfering with native biochemical processes, is bioorthogonal chemistry. This concept produced fantastic things, and I expect Professor Bertozzi to get the Nobel Prize within the next few years. The next one I want to mention is CRISPR, which the first work is something like CRISPR took place in the 1980s. The term was introduced in early this century, but Doudna and Carpentier produced their first paper on effective gene editing in 2012. I'm unhappy with them, Doudna and Carpentier. They stole our Nobel Prize. It's our Nobel Prize in chemistry. They deserve a Nobel Prize, definitely, but in medicine and physiology, not in chemistry. It's a phenomenal accomplishment, and we talk here about it. And finally, I want to mention click chemistry. The concept was first mentioned in 1998, but introduced to everybody, to all of us, in 2001 in the paper in the Angewandte Chemie by Sharpless, Finn and Kolb. Sharpless, in this very year, 2001, got a Nobel Prize for chiral oxidations and selective oxidations. Finn and Kolb are also excellent chemists. They came up with the paper, which let me describe very briefly. Usually, you have to synthesize a compound with specific, very well-defined structure. And if it's so, you have to do it. You have to produce carbon-carbon bonds, skeleton, stereochemistry. You end up with this specific structure. However, very often, perhaps even more often, you don't care about the structure. You care about properties. Properties, viscosity, melting point, solubility in water. And then you shouldn't be concerned with type of chemistry. Anything goes. Maybe even ugly chemistry is acceptable. And the result is that, first, before I move further, let me offer you two quotations. One was present in this paper by Sharpless and the rest in Angewandte Chemie. The most fundamental and lasting objective of synthesis is not production of new compounds, but production of properties. Great George Hammond said. And I found in one of the reviews, somebody says about, quotes Sharpless, reactivity isn't everything. It's the only thing. So reactivity is a very important factor, but let's think what are requirements of the click chemistry. It's a very special club, click reactions. So in this paper, they think about, they say we need to introduce chemistries which are simple, easy, and always successful. And they offered certain reactions, which I'll talk about later, but first, the click chemistry processes, the name is phenomenal. The idea is you mix reactants and here, yeah, click, and the reaction is over and the crystals show up. But those are the elements of the click chemistry. The yields must be very high. They should be quantitative if possible. There should be no byproducts. I want to emphasize this point because I return to it. They mention no offensive byproducts, but essentially it should be, byproducts should not happen. In other words, there should be additions, not condensation. Condensation, if they are condensations, they should produce only water or something like this. Chromatography is out of question. Crystallization, distillation are acceptable. the reaction must be stereospecific. They don't have to be enantioselective, but must be stereospecific. The conditions must be very simple. There should be no solvent, or solvents should be such as water. Water is acceptable solvent. The reactant must be really available, products must be stable. Important, the driving force for the reaction must be high, at least 20 kilocalories per mole. There must be reactions which happen on their own, without our help, at normal, reasonable pressures, reasonable temperatures. It's not a requirement, but it turns out that practically almost all, or maybe all, click reactions form carbon heteroatom bond, carbon sulfur bond, carbon oxygen bond. Synthesis is more interested in carbon-carbon bond formation. Here we form rather carbon heteroatom bonds. And finally, these click reactions should be tolerant of water and air. And at first, the group of Sharpless, Finn, and Kolb introduced several reactions. One became exceptionally popular, and is by far the most popular chemical reaction. It's a reaction between azide. I don't want to enter too deeply into chemistry. Azide is a group consisting of 3 nitrogen atoms in a row and a triple bond. This is cycloaddition. There are no byproducts, and such a ring is formed. This reaction was discovered in the 1960s by Huisgen, who is still alive, is I believe 101 years old, lives in Holland, and possibly, and if it's real, may get still a Nobel Prize. Maybe he deserves it. Anyway, this reaction is a great reaction. Fast, easy, simple products. The product looks ugly. From the chemist's point of view, lady or gentleman would never work with this kind of products. But who cares? We connect R1 and R2. Click chemistry about connecting molecular units, because that allows to produce properties. There was a problem with this reaction initially, because if you react these two, you end up with 2 products. This R1 can be either at this nitrogen atom or at this nitrogen atom. There are 2 products. But a year later, Sharpless on one hand and Meldal in Denmark on the other hand came up with the great catalyst, copper-1. Copper at the oxidation state of 1 is a great catalyst of this reaction. So copper-assisted or copper-catalyzed azide acetylene condensation is the name of this copper-1. Usually it's formed from from copper sulfate and ascorbic acid reduces copper-2 to copper-1. Great reaction. The yields are very often 100%. There's one problem with this reaction, even when catalyzed by copper. The problem is that it cannot be performed in living organisms. Copper is a poison. So how do we avoid copper if we have to avoid? Usually we don't have to, but if we have to avoid, Professor Bertozzi and Boons in Atlanta came up with 2 independent, different compounds, but the same concept, idea. Let's start with cyclooctane unit containing a triple bond. These 2 carbon atoms have the hybridization of sp, meaning this angle should be 180. So there's a tremendous strain in this molecule. So even without copper, it tends to produce cyclooctene, which is, the strain is much less. So this is the driving force to form this cyclic triazole compound. So those are 2 probably most popular click reactions. And other ones, reactions between thiols and double bonds. Thiols and double bond, thiol and triple bond, and particularly thiol with the double bond which is kind of activated. It's the Michael addition. EWG means electron withdrawing group. So you have, say, alpha beta unsaturated ketone, alpha beta unsaturated amide here, alpha beta unsaturated sulfone. Sulfur attaches to the atom further from the electron withdrawing group. Hydrogen attaches to this atom, and it's again addition. no byproducts. Another reaction which often is included in the category of click reactions is Diels-Alder reaction, but usually not carbon-carbon, but say carbon-sulfur system. Often people use reverse electron density reactants for this Diels-Alder reaction. Those are the main types of click chemistry. Recently, in the last maybe 5 years, Professor Sharpless promotes this very reaction. He's very proud of it. Suffix, sulfur fluorine exchange. I'm not fond of this chemistry because it produces a byproduct which is fluorine containing. It's not detrimental in some cases, but often it is detrimental. However, the reactions are fantastically fast, yields are excellent. There are good reasons to promote these reactions. So let me show you the next slide. I want to show that I've heard about click chemistry. I organized a symposium within ACS meeting in Anaheim some 7 or so years ago with my friend ZJ Witzak. And the result of the symposium was a book. Sharpless attended the symposium. We invited him and surprisingly did come, have a wonderful talk. But the reason I'm talking about it is that you see here 20% off. It's 20% off. If you wait until the end of this seminar, you get 20% off. And I believe 20% of 0 is really a very low number. So I encourage you to wait until the end of this talk, which will happen soon. So let me give you some examples. I want to talk for too long. I just want to give you some spectacular examples of applications of click chemistry. Here, some Korean scientists attached these molecules to surface of gold. Thiols form monolayers on the surface of gold. It's known for 30 years, more than 30 years. So the product is parahydroxyazobenzene, parahydroxy, and this is the azo group, paroxyazobenzene. Protonation and some electrochemistry cuts off this upper aromatic ring. We end up with the aminophenol, para-aminophenol. Oxidation produces this interesting quinonamine. Note an H group, oxygen, oxygen-carbon double bond, and in alpha-beta position is a double bond carbon-carbon. It's an object of the Michael addition. So now we can add a protein into it. One of the 20 amino acids present in this important proteins is cysteine, which has SH bond. This SH bond would not get into gold, but can get here. The result is the formation of the aromatic system after the addition of thiol here. So we end up with, so that's the driving force. But the result is immobilization of a protein. It can be any protein on gold. Very spectacular, I think, chemistry. Let me show you another spectacular chemistry. I found it in the paper in a review by Stoddard, but I think some Chinese chemist offered this wonderful topochemistry. This is the compound. You see there's a chain. At the end you have two objects of click chemistry, this cyclic triazoles. Via those triazoles, these dumbbells were attached. This benzene ring with terbutyl group. So it's sterically large and it makes impossible this chain to go through this ring. We have this pyrene pendants here and the same thing on the other side. The charge is 4+ in the upper product construct, 1, 2, 3, 4. And it turns out that this, say, ring, which is equivalent of cyclodextrin ring, is attached to this nitrogen atom. After using this base, it's deprotonated, and the result is that this and these molecules move towards the charge at triazole. Same thing here. The result is that now these pendants have ability to move, and they form the charge transfer complex here. They can get closer to this unit, and the result is the formation of this wonderful molecule, really wonderful molecule. Trifluoroacetic acid will protonate it and return back the original situation. I believe it's spectacular chemistry. Another example worth having a brief look, I'll be very brief, is this hydrogel polymer and this green asterisks or green stars get attached to the hydrogel via thiol reaction. And since this green thing is fluorescent, this has the product. This is the picture of the products, offering a beautiful color. There are hundreds, maybe thousands compounds meant to attack various cancers which contain click chemistry units. Let me give you one interesting example. This is the cancer cell. Without entering the details, it's decorated with azide system and 3 bonds. And this azide, via chemistry we talked about, cyclooctatriene, gets attached to this azide, and the result is attachment of chlorine E6 inside these capsules. Chlorine E6 is a photosensitizer with very strong anti-tumor properties. So it still requires irradiation, but after irradiation, you can see perhaps results. Anyway, the point is that now these capsules are very close to cancer cells. They are not anywhere in the system. They are where they are supposed to be. And here's the result. The mouse treated with saline, it's 0 days, you see the cancer, the tumor increases with time. But when treated the way we just discussed, the tumor almost disappears after 21 days. I think in one of the recent issues of Chemical Engineering was, there's a big title, First Click Products in Humans. Let's briefly see what it is. Sodium hyaluronate is modified with the presence of this kind of compound, which is a diene. It's a very effective diene for click chemistry. And doxorubicin, which is tetracycline of this type, is decorated with a variety of things, including TCO, trans-cyclooctane. Trans-cyclooctane is also strain containing cyclooctane ring. It produces the reaction, the Diels-Alder reaction between this diene and this N, allows for the formation of the adduct. The point is, as previously, sodium hyaluronate essentially somehow attaches to the cancer cells, and then the reaction to produce the click product forces the effective anti-tumor agent to act very close to the tumor cell. I'm a carbohydrate chemist, I used to be. Let me show, this is this sugar unit. It's trisaccharide containing a triple bond and azide. Here, unbelievably, the yield is 80%. But it's not just normal reaction. It's the reaction to produce this strange ring. Large ring is not 6-member ring. It's a huge ring. So the yield of 80% here is very, very high. Now, let me show you a picture of commercial drugs containing this unit, this triazole unit. But these are not drugs made via click chemistry. It turns out that this triazole unit reminiscence the amide bond, the amide system. It's isosteric with amide. So for many drugs are produced containing this triazole system. And the nature is easy to be fooled and believes it's an amide, and these drugs offer often fantastic properties. Here are a few drugs, but believe me, there are thousands, if not dozens of thousands of drugs which contain various units, click units, like here. Here they are mainly these cyclic triazole units. There's one problem with click chemistry. All those reactions are not reversible. In 2011, Bielski in Texas said that he can reverse click processes, but it turned out to be BS. It was not the case. But recently, Anslin offers very interesting process, which is type of click chemistry. And as you see, you can recover this reactant. The reactants you attach here is amine and R prime thiol. You can recover both thiol and amine. The only problem with this method is that it produces methyl sulfide. Trust me, it's the worst compound known to humanity. It smells like hell, or worse. However, it is a very elegant, phenomenal method. I just want to mention here that some years ago, together with my friend, we proposed another approach to situations when you have to, later, after everything you wanted to do is done, you want to split, disconnect the molecules. We propose to use a click chemistry between X and Y and W and Z, so that way you connect the circle with the square, and after the procedure is done, you can split them using the sacrificial unit. A and B is something which can be hydrolyzed or split using some very exotic chemistry, which will not do anything else, but just split the circle from the square. That's all I wanted to tell you. In conclusion, I would like to express hope that you learned something, and even those of you who don't perform a lot of chemistry, you should realize that click chemistry allows to change properties, to produce properties, also change properties. So if you have a polymer which is slightly too viscous, maybe a little bit of click chemistry will allow to decrease viscosity. If your compound, your polymer, monomer, whatever, is not water soluble, you can add something to it via click chemistry. There are other methods, but click chemistry is by far the best. So keep in mind that click chemistry is something which will let you change properties of products you're dealing with. Thank you very much. I see no reason why you should be crying. Why are you all crying?
Thank you, Roman. Let's open it up for any questions.
I try to respond to any questions.
I'll ask one, Roman. We were going to type them into chat, but we didn't get as many questions yet as I'd hoped. I very much admire the work of Dr. Carolyn Bertozzi, and I'd like to know what you think her greatest accomplishments are from using click chemistry to determine glycochemistry on cell surfaces. What do you think is some of her greatest accomplishments?
The greatest accomplishment, if I'm concerned, by far, is the concept of bioorthogonal chemistry. And originally, she used Staudinger ligation, which is not as effective as further click chemistries. And she used later on click chemistry. But my point is the concept. It's a phenomenal concept. It changed. It's a revolutionary concept. The results are fantastic. Some of conclusions she came up with. But the point is the concept of looking at the chemistry happening in the living biological organism. That's great. For some reason, nobody did it before. Did I answer your question?
Well, yeah, I think that's great. But what do you think is her greatest contributions to determining cell surface chemistry using click chemistry?
We learned tremendously much. I'm not able to answer the question, to enter the details. I think we learned about the surface of cells so much, but I cannot tell you which one is the particularly important. I don't know, you know better.
Yeah, okay. Are there other questions for Roman?
Stop your tears. Start asking questions.
I'll ask one, Roman.
Huh?
It's always amazed me how nature can create the most marvelous compounds at room temperature, in air, with water, with 100% yield, or seemingly 100% yield, whereas we chemists have to do so much with much harsher conditions and get poorer yields. Do you think nature practices click chemistry?
That's a wonderful question, but John, let me disagree with you. On one hand, you're absolutely right. The yields in nature are always 100%, and nature always produces the desired compound. However, nature is very, very wasteful. Let me give you an example. Nature, to produce an enantiomeric pure compound, starting, let's say, from the non-chiral compound, uses enzymes which contains hundreds, if not thousands of atoms. We learned that chemists cannot produce 100% pure, enantiomeric pure compound, but our methods are much simpler. Noyori and Sharpless came up with just, I don't know, 50 atoms in the complex to produce almost, not as good, but almost as good compounds. Let me look at immunology. Nature is totally wasteful. The number of atoms, the time, nature is not that good. Now let me return to your question about click chemistry. Yes, the nature does use sometimes, not the Huygens reaction, Huygens cycloaddition, but some Michael addition of thiols to unsaturated carbonyl compounds. Yes, the nature uses those reactions. So the nature learned about the click chemistry earlier, before Sharpless.
Very good. How about industry? Is industry practicing click chemistry?
Very much so. There are polymers containing click chemistry, thousands of them. People spend too much money and too much time, but some of them are wonderful. The click chemistry, if you want to attach something to the surface, so you use click chemistry to surface of a cell, to surface of a particle, capsule. So industry uses click chemistry. There are two areas, I would say, particularly important. One, tracers, radio tracers. You have a compound which contains an element with half-life of a few hours. So you need to hurry up with your chemistry. So click chemistry is wonderful because soon after the product was produced, the element, radioactive element was produced, you have it attached to whatever you wanted to, and you can run the test on the patient. Other tracers, fluorescent tracers, and so on. So the diagnostics industry uses a lot of click chemistry. Now, therapeutic industries is starting using chemistry. We saw the first compound, which will be used in humans, in one of the slides, but animals, perhaps we care less. We couldn't care less so much about animals as we care about humans. So there are some drugs containing click chemistry for animals. And then polymer industries using more and more click chemistry.
Any other questions for Roman? Roman, I'll ask you a question that appeals to your carbohydrate chemistry background. In the past, I'm a biochemist, as you know. There has been the age of the nucleic acids, RNA and DNA, and proteins and lipids. Do you think that the next few years will be the age of glycobiology as a dominating force?
Great question, thank you. Absolutely, absolutely. We thought the sugars were not important. The sugars attached to anything are very important. The structure is very important. And the process started some 30 years ago, I would say. Rob Danishevsky was the first who synthesized this very complicated oligosaccharides. And so we are not only able now to determine the structure, but also to produce these oligosaccharides. So I expect that synthetic chemists will be able to produce to full nature with modified glyco structures like this, where sugars are attached to proteins, to nucleic acid, and so on. I think is the next step, maybe the most important step, even more important than nucleic acids or protein and so on.
Further questions for Roman?
Roman, can you hear me?
Yes, I can. Hi, Mike.
Hi. You described the click chemistry as something of very broad application, and yet it seems to me that the reactive groups involved are not the kind of groups that you often encounter in natural compounds or compounds in general. Can you comment on that?
You're absolutely right. Most, let me put it this way, not yet. Because more and more we learn that this click chemistry products are benign and don't hurt nature. But it takes time to prove that they really don't, that there are no any result concerning byproducts or effects within the body. So what I'm saying is 20 years is a lot of time. It's almost 20 years since the beginning of click chemistry. But in terms of medicine, it's not that much time. Give me 20 more years. I won't be around, but click chemistry will be around in many, many drugs.
Thanks.
Anyone else? Well, if not, I hope we can all thank Dr. Roman Bielski and Dr. John Newport for introducing him. And I wanted to remind you all that on December 2nd, we are hoping to have a talk on COVID and its effect on consulting by a person from the Delaware Public Health Division, Dr. Rick Pescator, who will be hosted by our own Fred Hartman. And with that, I think we can all go about our evening's business. And thank you very much, Roman and John, for this. Thank you for this fine talk, Roman.
Thank you.
Thanks, Roman.
Thank you, Roman.
Thanks.